Roots are essential for anchoring plants into the soil and absorbing water and nutrients, but their effectiveness can be greatly enhanced by symbiotic organisms. Among the most important ones are arbuscular mycorrhizal fungi, or AMF for short, which form an association with an estimated 80% of plant species. They act as extensions of the roots, absorbing vital nutrients, particularly phosphorus, and play a key role in supporting plant health and productivity. Quantifying AMF in crop plants and assessing how they respond to factors like agricultural practices, soil conditions, and plant genetics is crucial, given their significant role in plant growth and health.
The conventional method for AMF quantification is microscopy, which involves staining the roots and meticulous observation under a microscope. This is time-consuming and subjective because results depend on the observer's skills and experience. We have validated the use of absolute quantitative polymerase chain reaction, or absolute qPCR for short, to quantify AMF in the roots of different crops. This high throughput and reliable technique is versatile and suitable for a wide range of research projects. The process is divided into two parts. First, the sample preparation, and then the qPCR. Let's look at the protocol for part 1, sample preparation. First, we must obtain standards. This can be either a clone fragment from a known species or a synthetic DNA fragment that contains the target sequence. Next, we collect the samples. Roots are collected, washed, and stored in the freezer. Then, the samples are ground. This can be done by placing them in a 2 ml reaction tube with 2 mm metal beads. Freeze for 2 hours at -80 and grind them with a tissue. The final step is to extract the DNA. Weigh the exact amount of ground roots and extract the DNA, for example with a plant DNA kit. Now that the samples are ready, we can move on to part 2, the qPCR. The first step here is to quantify the DNA concentration. We can do this using a plate reader. If necessary, dilute the samples with molecular grade water. Next, we will prepare the qPCR. Each sample is measured in triplicates. Include a dilution series of the standard as well as negative controls. In a final volume of 50 microliters, add 200 nanomolar of each primer. In our case, we chose the primers AMG1F and AM1. Add 1.5 microliters of DNA template and your enzyme of choice. In our case, we use the kappa cyber mix. We are ready to run the qPCR. The qPCR program that we will use is initial denaturation for 3 minutes at 95 degrees, followed by 40 cycles of denaturation at 95 degrees for 10 seconds, annealing at 62 for 30 seconds, and elongation at 72 for 20 seconds. Finally, we will do a melting curve analysis from 55 to 95 degrees. Note that the program needs to be adjusted if using a different enzyme. Finally, we can quantify the AMF in our samples. Using the standard, a regression line is calculated to determine the number of gene copies present in each sample, for example, using the program which goes with the qPCR machine. You have now quantified AMF using absolute qPCR.
Using absolute qPCR to quantify the AMF in root samples allows for the processing of a huge number of samples at the same time. Furthermore, it comes with the advantage that it does not rely on the observer's subjectivity, as happened with microscopy. It also has another advantage. We can pair these results with the sequencing of the AMF diversity. One weakness of this method is that it cannot differentiate between the different AMF structures, as happened with the microscopy. In addition to this, it has been reported that this method yielded inconclusive results for specific crops. Therefore, we recommend you to validate this qPCR approach for your specific crop.
More information on how we used qPCR for AMF quantification can be found in our publications, and more information on related findings from the Root to Rest project can be found on our website, or you can subscribe to our channel.